Thanks everybody. My today's lecture is about male reproductive system and I am your coach Dr. Mudassir. Without wasting any time, let us begin. Male reproductive system mainly consists of a pair of gonads and the ducts which carry the spermatozoa or the germinal cells produced from the gonads are epididymis, vasa deferens, ejaculatory duct and urethra. It also has accessory glands like seminal vesicles, prostate gland and bulbourethral gland. All these structures, the ducts, the accessory glands and gonads are supported by spermatic cords, scrotum and penis. Now, before understanding the physiology, we should understand the functional anatomy of testis. So, first and foremost, its location. These gonads or testes are basically ovoid structures present in the scrotal sacs. They are covered by three layers. The innermost is tunica vasculosa, the outermost then next is tunica albuginea and tunica vaginalis. This tunica albuginea divides the testicular cavity into several lobules. You can see here that tunica albuginea it divides the testicular cavity into several lobules by the fibrous septae which are running from tunica albuginea to the interior. Now, these lobules, what do they contain? Let us see. The components of each lobule is seminiferous tubular component and interstitial fluid compartment or interstitial component. Now, you can see here each lobule in its cut section looks like this, with central tubular component which is called as seminiferous tubular component and periphery surrounding it is interstitial compartment or component. Now, we will see what are the components of the seminiferous tubules. Seminiferous tubules are basically covered by a capsule which consists of fibroblasts, myoid cells. Inside this capsule is basal lamina, over which are cuboidal cells which are stratified or arranged in a stratified manner. That is why this epithelium of seminiferous tubules is called as cuboidal stratified epithelium. This cuboidal stratified epithelium mainly consists of two types of cells. The germinal cells, which are cuboidal cells, which we can see here in different phases of development, and the larger pyramidal cells which are called as Sertoli cells. Now, these Sertoli cells play a very important role. The first important role played by them is providing support and nutrition to the growing spermatozoa. Next, they remove the byproducts of spermatogenesis by phagocytosis, and then they maintain a blood testis barrier. What is this blood testis barrier? This blood testis barrier is a barrier formed between the seminiferous tubular fluid and the blood. So, this barrier is, this is the barrier formed between the seminiferous tubular fluid and the blood, which is formed by the tight junctions. You can see here these are the two subsequent or adjacent Sertoli cells. On the basal side, these are the tight junctions. So, the tight junctions between the two adjacent Sertoli cells form a tight junction which is called as blood testis barrier. So, what is this blood testis barrier? By definition, it is a barrier between seminiferous tubular fluid and blood formed by tight junctions between Sertoli cells. Its main function is to protect the germ cells from blood-borne noxious substances, prevent antigenic products of spermatogenesis to enter the circulation, and maintain the composition of seminiferous tubular fluid. The other important function observed by Sertoli cells is secretory function. It secretes seminiferous tubular fluid, Mullerian duct inhibitory substance, inhibin, androgen binding protein, estrogen and transport proteins. Mullerian inhibitory substance inhibits the development of indifferent gonad into female reproductive system in male fetus under the influence of Y chromosome. Next important substance is inhibin. Inhibin inhibits spermatogenesis up till the age of puberty, and during adult age it exerts a negative feedback inhibition on secretion of follicle stimulating hormone from anterior pituitary. Androgen binding protein is another substance secreted from Sertoli cells which maintains high concentration of testosterone, which is required for spermatogenesis inside the seminiferous tubules. Next component of each lobule is interstitial component. Interstitial component has got three things. One is interstitial cells of Leydig, blood vessels and fibroblasts. You can see here these are the interstitial cells of Leydig, blood vessels and fibroblasts. Interstitial cells of Leydig play an important role by secreting androgens. Androgens are testosterone, dihydrotestosterone and androstenedione. Now, going beyond this, we will see what how this seminiferous tubules, what are the other aspect of seminiferous tubules. Now, these lobules, as I was discussing earlier, it contains seminiferous tubules and interstitial compartment. Now, these seminiferous tubules, if you see carefully, you can see here it has got two components. One is convoluted component, you can see here it has got convolutions here, and the straight part. The convoluted part leads to straight part. The straight part on the mediastinum of the testis joins to form retrotestis, and this retrotestis again gives away efferent ductules. These efferent ductules fuse to form epididymis, which marks the beginning of ducts of male reproductive system. The epididymis is a long tube which passes from the scrotum into the abdominal cavity, and behind the urinary bladder and in front of rectum it joins with the duct of seminal vesicles, another accessory gland, to form ejaculatory duct. This ejaculatory duct passes through the prostate gland and fuses with the urethra. After some point of fusion of ejaculatory duct with urethra, in the distal part it is joined, prostatic urethra is joined by bulbourethral glands duct, which secrete mucoid substance, which provide lubrication to spermatozoa. So, that was about the ducts and accessory glands of male reproductive system. This finishes about the structural component, functional anatomy of male reproductive